We're on Unit 6, Gene Expression and Regulation, and we're going to be doing Topic 2 today. Let's remember our overarching big deal here, that an individual's genotype is physically expressed through their phenotype. Today we're going to describe the mechanisms by which genetic information is copied for transmission between these generations. And our notes are going to start on page... Here we go, 14. So let's get into it. We're going to talk about DNA replication. So up until this point, we've talked about mitosis and meiosis, cells growing, cells dividing, sort of crossing over of these genes. We've talked about Mendel and his alleles. We've talked about dominant and recessive. But the big question here is, how do these phenotypes, how are they actually expressed? And one of the first things that we do here, right, through mitosis and meiosis during the S phase is our DNA replication. So it occurs during the S phase of the cell cycle. Remember, S stands for synthesis. We are synthesizing a new piece of DNA. And our big question today is, how does DNA replicate? It's an interesting process. There's a lot of key players, so I'm really excited to get into it with you guys. Before we get into it, we didn't know exactly how DNA sort of passes down its genes. So there's 3 different modes of DNA replication. These are 3 alternative models for DNA replication that scientists came up with in the past. The first is a conservative model. So this is the idea that you have your original parent DNA here. We model it in gray, and that it separates. A new copy is made of each strand. Those new copies come together, and then the original one is conserved through those generations. So that's our conservative model. The next one over here is our semi-conservative model, for where you have your parent cells up here. They're both represented in gray. The DNA will kind of split. A new copy will be made based on the old copy, and half of it gets passed down to one generation, another half gets passed down to the other. We call this semi-conservative. And finally, the dispersive method, which is this idea that everything kind of just gets mixed up. It's an interesting idea. I don't know if it holds too much weight. What do you guys think? So the conservative model here, the parent strands directly synthesize an entirely new double-stranded DNA model. So that's what we sort of mentioned earlier, that you have your parental strands up here in gray. They separate, new piece of DNA is replicated, and now we have a new strands of DNA separate from the parental strands. The parental strands are fully conserved through these generations. The second model here is the semi-conservative model, where 2 parental strands each make a copy of itself. So they separate, one makes a copy here, and that's represented in the blue. And after one round of replication, 2 daughter molecules each have 1 parental strand and 1 new strand, as you can see here, represented by the gray and the blue. And then finally, we have the dispersive model. These are the material in the 2 parental cells is like randomly dispersed between the 2 daughter molecules. So you can see here the parental strands up top, and then the blue is down below. After one round of replication, the daughter molecules contain a random mix of the parental, again in gray, and the new in blue. So of these 3 models that we looked at, what do you guys think? Which one do you think is the real way that we replicate this DNA? You can pause here, discuss it in class, or if you're at home with a pet. Awesome discussion. Which model is correct? Well, these dudes here in 1954, the 50s were a really big time for DNA discoveries. These 2 guys, Messelson and Stahl, performed an experiment using bacteria, and it's actually like a really beautiful, nicely designed experiment. So what they did was they took bacteria and they cultured it with this heavy isotope. This is nitrogen 15. It's heavy, it's denser than the other isotope, which is N14, right? So they cultured this bacteria with this heavy isotope in nitrogen, which is a main component of bacteria, and then they put it into culture, into a medium with this lighter isotope. and the DNA was centrifuged and analyzed after each replication. So after each replication of the DNA, they went in, they centrifuged it, and they took a look at how this density kind of separated these two molecules. And here's what they found, which, by the way, if you saw semi-conservative as the most logical model here, you were right. By analyzing these samples of DNA after each generation, it was found that the parental strands were following the semi-conservative model. So you can see in the first, before any replications happened, it was 100% this heavier N15 isotope. After the first replication, you can see here, it's a little bit higher, and then it starts to split 50-50, 75-25, that sort of thing. So by seeing this evidence, they said, oh, it has to be the semi-conservative model. And this is, I like this chart over here on the right because you can see the parental strands start in red. This sort of helps to explain how after the second generation, everything kind of gets smaller and smaller from that original N15, is that you have your two parental strands up here in red. One of them is replicated down here in the first generation. The new strand is now in orange. Same thing on this side. So you have the red, new strand is in orange. But once you get to the second generation, this orange strand in the first generation becomes the parental strand for one of these here. So if you track the original red, you can see it gets smaller and smaller and smaller. The percentage of it, I should say, gets smaller and smaller. Really interesting, beautiful experiment to kind of just put a pin in this data that they're seeing. Very cool stuff. So how do we replicate DNA? Here it is. This is everything you need to know. These are all the players that are involved. There are a lot of players. You see some ASEs, some enzymes here. You see a leading strand. We see another ASE, a DNA polymerase. We see a fork. I hope you're hungry because we got replication forks. So here, let's take this step by step. So DNA replication begins at the site called the origin of replication. Kind of, a no-brainer. Like, take solace in the fact that biology makes sense now. The origin of replication is where replication originates. Origin of replication is where DNA replication originates. Various proteins attach to this replication fork and open the DNA. Oh, I'm sorry, origin of replication and open the DNA to form a replication fork. This is your replication fork. Think of it like a fork in the road. Interestingly enough, our DNA could have multiple replication forks, right? Your DNA is so long, if we started at one end and just went to the other end, it would take too long. So there's multiple replication forks that open up on a single piece of DNA. Interesting stuff. It's like a fork in the road. Okay, so replication fork is open at the origin of replication. Love it. Helicase is the first enzyme we're going to see here. Helicase is going to unwind the DNA strand at each replication fork. The next protein, the next player in this that we're going to see is called a single-stranded binding protein. These do just what the name says. They keep the DNA from rebonding to itself. They bind to the single strands of DNA. Single-stranded binding proteins bind to the single strands of DNA, and it helps keep it open. One of my favorite science words of all time, topoisomerase. This is the enzyme that helps prevent strain ahead of the replication fork by relaxing supercoiling. And I'll do this for you guys as a model when I get back into the classroom. If you've ever taken like an elastic band or something like that and just kept twisting it and twisting it and twisting it and twisting it, eventually it's going to kind of twist in on itself. That's what we call supercoiling. And if your DNA is being pulled apart, then the supercoiling could damage that DNA ahead of where the replication fork is. So this enzyme comes in and sort of relaxes it, make sure that it doesn't snap. So this is what it looks like. We have helicase that comes in. We have our replication fork at our origin of replication. Helicase comes in to help unwind that DNA strand. Single-stranded binding proteins come in and bind onto their proteins that bind onto the single strand of DNA and help stabilize it. And finally, topoisomerase comes up ahead of the helicase. We call this upstream. Downstream, upstream. We're always swimming upstream. We're like salmon. We swim upstream. The helicase goes upstream. So the topoisomerase is upstream of the helicase, and this is just to make sure that the DNA upstream doesn't get damaged or over twisted or anything like that. So this is steps 1 and 2. Step 3, here's another player in the game. So far we have helicase, we have topoisomerase, and we have single-stranded binding proteins. The next player in this game is primase. It's an enzyme, and it initiates replication by adding short segments of RNA. We call these primers, and we add those to the DNA strand. Now, it has to be short segments of RNA instead of short segments of DNA because it's a primer. They have to be removed at some point. If it was short segments of DNA, then the enzyme that comes and removes it might mistake it for just part of the replication process. So primase comes in, we add short segments of RNA, we call those primers. These enzymes, the enzyme that synthesizes DNA can only attach new DNA nucleotides to an existing strand of nucleotides. So the primer also acts as a starting point, as something for these other nucleotides to attach to as we're putting together and building the strand of DNA. They serve as a foundation for DNA synthesis. Sorry, there you go, pause to take notes. And here's what it looks like. So we have our replication fork at our origin of replication. We have our helicase right up top. Oops. We have our topoisomerase that's upstream, making sure that things don't supercoil. And we have our single-stranded binding proteins on either of the single strands to ensure that they don't, coil back up again. So next, an RNA primer is put onto the strand of DNA, and it's put on either side. We're going to represent it right here. Note the directionality of your DNA. You got your 3' end up here, you have your 5' end over here. So I mentioned this. I mentioned the directionality of this is because DNA polymerase will come and attach to each of the primer on the parental strand, and it moves in the 3' to 5' direction. It moves in the 3' to 5' direction. DNA polymerase 3 moves in the 3' to 5' direction. Say it with me. DNA polymerase 3 moves in the 3' to 5' direction. And as it moves in that direction, and I'm going to back up a little bit and show you. So it's going to move from the 3' to 5' direction. So from here, where the primer is, into the replication fork. Right? It moves in the 3' to 5' direction. And as it moves, it adds the new strand of nucleotides in the 5' to 3' direction. This is a weird concept to wrap your brains around, but think of it like you're walking down the street and you're dropping breadcrumbs behind you. As you're dropping breadcrumbs behind you, you're dropping it in the opposite direction in which you're moving. So DNA polymerase 3 moves in the 3' to 5' direction, but the new strand is added in the 5' to 3. Okay? It's weird to wrap your head around, but I found that the dropping breadcrumbs behind you analogy is like the easiest way to think of this. So the DNA polymerase 3 that follows helicase, the one that we saw in the last slide, is known as the leading strand. It only needs one primer, right? So you toss the primer on, the DNA polymerase 3 attaches to the primer, it moves in the 3' to 5' direction. So it's directionality, it's a one-way street, and it's going into the helicase. We call that the leading strand. On the other parental strand, DNA polymerase 3 moves away from helicase. It can only move in one direction, from 3' to 5'. So we call that the lagging strand. Takes a little bit longer, right? It's on the sloth hiking team like I am, right? It takes a little bit longer. It needs many primers, and it's not like one continuous go. So here's kind of what it looks like. Just to recap, we have our replication fork and our origin of replication. We have our helicase that's coming and splitting our piece of DNA. We have our topoisomerase that's making sure that things don't supercoil. We have our single-stranded binding proteins binding to the single strands, making sure it doesn't recoil. And we have our primers, one right up here at our 3' end. But you'll notice that down here on our lagging strand, you have 2 primers. If you can like sort of imagine this, your 3' end is over here. So your DNA polymerase, so there's your leading strand, DNA polymerase is going to move inwards 3 to 5. No big deal. It can just like go on its own, right? So it's following helicase. As helicase is moving, topoisomerase is moving, DNA polymerase 3 is all moving, and they're all moving in the same direction. But on this poor little lagging strand here, DNA polymerase 3 again moves in the 3 to 5' direction, but in this case it's moving away from helicase, right? DNA is anti-parallel, so there's directionality to this, right? So you might be thinking to yourself, well, what the heck happens, right? How are we doing this? How do we have, excuse me, sniffles? How can we have these little segments down over here? So the leading strand, again, synthesized in one continuous segment, but the lagging strand, since it moves away from the replication fork, it's synthesized in these chunks. This is another one of my favorite science words of all times. We call it Okazaki fragments, named after the dude who found them. These are segments of the lagging strand, right? So you'll notice down over here, boom, there's your leading strand. Let's see that one more time because it's like, whoa. You have your helicase, you have your topoisomerase, you have your single-stranded binding proteins, your DNA polymerase 3 bound to your primer here, and away it goes. You'll notice it's moving from the 3 to 5' direction, but as it's dropping these nucleotides, it drops it in the 5 to 3'. Those are your breadcrumbs that you're leaving behind you as you go. Right, so you're moving in one direction, but you're dropping breadcrumbs the other way. Down over here in the lagging strand, we have the same thing. We have our primer, we have our DNA polymerase 3, but you'll notice that it moves in the opposite direction. So as we're putting down these nucleotides, again, we're moving in 3 to 5, we're dropping them in 5 to 3. We create these things called Okazaki fragments. And they're just these small portions of DNA. They're just these fragments of DNA. And they are separated by these RNA primers. Okay? But you might think to yourself, well, how are we going to get a full strand of DNA here? We have these RNA primers on here. So after DNA polymerase 3 forms an Okazaki fragment, DNA polymerase 1 replaces the the RNA nucleotides with DNA nucleotides. So 3 puts on the DNA nucleotides, 1 takes off the RNA, replaces it with DNA. Once those primers are gone, we have these little sticky ends, right? We have these small segments of DNA, but that backbone still has to be connected in there. You still have to put your left hand on the right shoulder of the person in front of you to form that backbone. So that's the job of DNA ligase, another enzyme. You can see ASE, and it joins these fragments together. They're kind of like stitching them together, and it forms a continuous DNA strand. So main players here, we got our helicase. Helicase is puttering along. Topoisomerase is following along with it. Single-stranded binding proteins are making sure everyone's stable. We have our RNA primers. We have our leading strand of DNA. And now we have our lagging strand of DNA down below with our little Okazaki fragments. So let's see what happens. So DNA polymerase 1 replaces those RNA primers with DNA nucleotides. Then ligase comes along and it starts connecting these fragments, right? So let's see that one more time. Hang on, going back. Right, so we got our primers. DNA polymerase 1 replaces the primers. Boom. See, once it was RNA primers, now it's DNA nucleotides. And then ligase will come in and connect the fragments together, making sure that the backbone's all connected and we got that one big strand of DNA. So this is putting it all together. This is what everything looks like. I'm going to post this video as well, the link to this video. It's an awesome sort of accompaniment to this lesson. It's from Bozeman Science. That's Mr. Anderson. I know that you guys like him. So if you have extra time, go ahead, watch that. So let's talk about some of the nuances that go on with this replication. So since DNA polymerase 3 can only add nucleotides to the 3' end, there's no way to finish replication at the 5' end of the lagging strand. It's a lot of information to kind of take in at once, but when we see it visually, it'll make more sense. Over many replications, this would mean that DNA would become shorter and shorter and shorter and shorter. Okay, so how are genes on DNA protected from this? There are these things, we've talked about it in class before, like in passing. We're going to talk about it a lot next year if you take biomed with me. But at the ends of the DNA, and this is like at the end of the DNA, not like a single segment, but at the actual ends. Because remember, our chromosomes, our DNA is linear. We have these things called telomeres. They are repeating units of short nucleotide sequence. They do not code for genes. Many people think that telomeres are the key to anti-aging because every time our cell divides, we replicate our DNA. Every time our DNA is replicated, you kind of got to come in and trim off the ends. If anyone here is a knitter or a sewer or something like that, usually like you'll make a product, you'll make something like a scarf, and then you'll have that little bit at the end that you got to snip off. That's your telomeres in your DNA. So we're like we're like weaving together this new piece of DNA, and then we just need to cut off the ends real quick, just to like make sure that everything's good. That's your telomeres. They don't code for genes. They get shorter and shorter every time your cells divide. They form a cap at the end of DNA to help postpone erosion of this DNA. So again, there's a ton of scientists out there who are doing research in this telomeres, what they do, how we can kind of, if we added on extra sticky ends on the telomeres, could we reverse aging? Like, it's a huge business. Very interesting stuff. The enzyme telomerase adds telomeres to DNA, right? So just bump up your telomerase, I guess. No more wrinkles. Okay, so we have to proofread, right? So we're taking this piece of DNA that's millions and millions and billions of base pairs long, and we're replicating it one by one. We're going A to T to C to G. We're going to have to proofread it. We're going to have to repair it. As DNA polymerase adds nucleotides to the new DNA strand, it proofreads the bases that are added. If an error occurs, a mismatch repair will take place. Enzymes remove and replace the incorrectly paired nucleotides, and if segments of DNA are damaged, nuclease, which is an enzyme, ASE, can remove segments of nucleotides, and DNA polymerase and ligase can replace the segments. So this is essentially like, it's a cut and paste. DNA polymerase will add the nucleotides, and as it's adding, it proofreads, right? It'll go back, it'll read its work. If errors occur, and it's like a simple error, we can literally just replace it on the spot. If bigger errors occur, then we come in, we snip out the whole piece and replace it. So let's do, pause here if you want to take notes. Let's do a quick review. So if parental DNA strand reads 5' ACGTAC to 3', what would be the newly synthesized complementary strand read? Please do the primes in there as well. So pause here, take a moment, do the review. Congratulations, you got it right. So you would do the 3', and then A goes to T, G goes to C, blah, 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 all the way down. Make sure you add your primes in. This is very important when we're talking about DNA replication. Quick review number 2. Summarize the role of helicase, topoisomerase, and DNA polymerase 3. Pause here to take a minute and answer this question thoroughly. Correct. Helicase will unzip the DNA. Topoisomerase reduces that strain upstream. DNA polymerase 3 adds nucleotides to the new strand of DNA. Congratulations, you're a genius. All right, what direction does DNA polymerase 3 move along the parental strand? Take a minute, pause, do this. Correct. It moves from the 3 to 5'. It moves from the 3 to 5' direction. DNA polymerase 3 moves 3 to 5. You can even think of it as DNA polymerase 3, 3, right? Because DNA polymerase 3 moves from 3 to 5'. That's the way the physical enzyme moves. Why do Okazaki fragments occur on the lagging strand? I wonder if it has something to do with the question above. Pause here, answer this thoroughly. It's because the fragments of DNA occur because 3 moves away from the replication fork. It replicates in chunks. It moves from the 3 to 5 prime direction. Fantastic. God love it. Is that it? That's it? All right, awesome. Thank you guys so much. I'm going to post that video. Hope you have a lovely day. I'll see you in class.